When I first came to Stanford, this new sequencing technology was just being implemented, so there was one machine with rather low output. So one of the biggest challenges for us was, in fact, the initial processing of human genome sequences coming off the sequencer. So we need to get the reads, map them, and call the variants, and that was a very time-consuming process. This would take us 36 hours with our existing system. So in fact, we did implement the Bina system, which we needed because the project we were doing with the VA had 400 genomes, and 36 hours with 400 genomes is over a year, as you might imagine. In fact, it's close to 2 years. And so the Bina system reduced it from 36 hours to about 5.5 hours. And so we're able to process most of these genomes in about 2 months. So it really did greatly accelerate things at an enormous level. In fact, we generate about 2 trillion bases a month, 2 trillion nucleotides of sequence every month from our center. So it's a huge amount of volume, and it's had an enormous impact then in 2 ways. One is we can now do clinical research sequencing, where we're sequencing patients like cancer patients or those who have a disease whose diagnosis hasn't been figured out. And the other is just taking on massive research projects. And so it is a big challenge. It's one where we are working with internal groups, the IT group at Stanford, as well as companies on the outside to help us do this in a more efficient and robust fashion. And the goal is to do this stuff cheaper, faster, and more efficiently, and in a more painless fashion, because it's a very fast-moving field, and mining large amounts of data is not a trivial task. I actually think genome sequencing will be routine in about 2 years, if not before, certainly for the cancer, and probably heading that way for the undiagnosed disease.